Hello everyone, my name is Renata. I'm a PhD student and my project is Mechanisms of Platelet Activation and Platelet-Lymphocyte Interaction in COVID-19. SARS-CoV-2 virus emerged in China in December 2019 and spread rapidly throughout the world, resulting in a public health problem with thousands of infected people and increase in the number of hospitalizations and deaths. With the advancement of research and creation of vaccines, the number of cases and hospitalization has been decreasing. However, COVID-19 is still in circulation. This year, the number of cases is around 400,000 and the number of dead is almost 3,000. Around 10 to 30 patients may develop the long COVID, which is characterized by the continuation or development of new symptoms within 3 months after the initial infection with SARS-CoV-2 and which persists for at least 2 months and cannot be explained for an alternative diagnosis. Patients with long COVID may present neurological symptoms such as difficulty thinking or concentration and headache, respiratory symptoms such as difficulty breathing and cough, digestive symptoms such as diarrhea and stomach pain. Furthermore, they can present circulatory abnormalities such as an increased risk of persistent thrombosis. It is in this scenario that platelets appear to play an important role in the pathophysiology of COVID-19. Some studies have already shown that platelets from patients with COVID-19 are more activated and hyperreactive. Activated platelets can interact with leukocytes, modulating their response. Our group has already demonstrated that the formation of platelet monocyte aggregates participates in thromboinflammatory response during COVID-19. Other studies have already demonstrated the platelet-neutrophil interaction resulting in the release of NETs, which, due to their prothrombotic characteristics, can trigger the state of immunothrombosis in severe patients with COVID-19. However, the platelet-lymphocyte interaction has been much less explored. Therefore, the general objective of this work is to investigate the mechanisms of platelet activation and platelet-lymphocyte interaction during SARS-CoV-2 and long COVID-19 infection. The first specific objective is to characterize the platelet-lymphocyte interaction in acute COVID-19 and in patients with long COVID-19. So to do this, we are going to perform a flow cytometry to identify the specific population of lymphocytes that interact with platelets. We are going to analyze 68 cryopreserved peripheral blood mononuclear cell samples from COVID-19 patients enrolled from 2020 to 2021, and samples from patients with a confirmed diagnosis of long COVID. The second objective is to analyze the platelet-lymphocyte interaction in an in vitro infection model. We are going to isolate platelets from healthy volunteers and infect them with SARS-CoV-2 or viral proteins. Then we are going to perform a co-culture with lymphocytes collected from healthy volunteers. By flow cytometry, we will analyze the population of lymphocytes that form aggregate with platelets and investigate whether the interaction between these two cells modulates the response profile or modulates a specific subpopulation of lymphocytes. The last objective is to investigate the mechanisms of platelet activation and platelet-lymphocyte interaction. To identify the mechanisms of activation, we are going to analyze the profile of phosphorylated protein kinases in platelets of volunteers diagnosed with long COVID using a human phosphokinase array. In a previous work, we described the platelet activation pathways in acute COVID-19, such as mTOR pathway. So now we ask if this pathway remains activated in long COVID. Furthermore, we also question whether this platelet activation through this pathway is related to lymphocyte activation. So we are going to perform a co-culture with platelet treated with rapamycin, that is an inhibitor of mTOR pathway, and we are going to do a co-culture with lymphocytes, and then we are going to analyze the lymphocytes populations through flow cytometry. Doing this research, we expected to elucidate fundamental mechanisms in which platelets are involved in immune regulation during COVID-19 and identify the subpopulations of lymphocytes that form aggregates with platelets. We believe that platelets may play a key role in modulating lymphocyte response to an exhaustion profile, and this study is important to understand the role of platelets as immune sensors and regulators, participating in the mounting of the adaptive immune response in viral infection and other diseases.